Renal handling of sodium. Sites of sodium reabsorption. Sodium is the major osmole in ECF and determines ECF volume. It is filtered and reabsorbed in the largest amount. Sodium is reabsorbed in all parts of the nephron except the descending thin segment. Sodium absorption across the basolateral membranes in all parts of the nephron is by the sodium-potassium ATPase pump, an active process requiring energy. Sodium reabsorption, therefore, consumes a tremendous amount of ATP and oxygen. Sodium reabsorption accounts for majority of the oxygen consumption by the kidneys. This is why the kidney receives approximately 25% of cardiac output. Mechanism of sodium absorption across the luminal membrane differs in different parts of the nephron. Reabsorption of water and many other essential solutes such as glucose, amino acids, calcium, chloride, lactate, phosphate are linked to sodium reabsorption. The amount of sodium reabsorbed in the PCT is 2/3 or 60%, 30% in the thick ascending limb, 7% in the distal tubule, and 3% in the collecting duct under the influence of aldosterone. Mechanisms of sodium reabsorption in different parts of the nephron. Proximal convoluted tubule, PCT. Sodium is pumped into the interstitium by the sodium-potassium ATPase pump in the basolateral membrane. Sodium is absorbed by secondary active cotransport or countertransport from the tubular lumen into the epithelial cells down its concentration and electrical gradients created by the activity of the sodium-potassium ATPase on the basolateral membrane. Examples of cotransport are sodium glucose, sodium amino acids, sodium phosphate, citrate, lactate symporters. Example of countertransport is sodium hydrogen antiporter. Water follows sodium due to osmotic force. Sodium reabsorption in the thin descending segment. There is no reabsorption of sodium in this segment. Only water is reabsorbed in the descending thin segment. Sodium reabsorption in the ascending thin segment. The sodium reabsorption in this segment is by active transport across the basolateral membrane and passive reabsorption across the luminal membrane. Sodium reabsorption in the thick ascending limb. 30% of filtered sodium is reabsorbed in this segment. Sodium-potassium ATPase pump acts on the basolateral membrane and sodium-potassium chloride cotransporter in the apical membrane. Furosemide or the loop diuretics act on the sodium-potassium chloride cotransporter in the apical membrane, causing loss of sodium, potassium, chloride, and water in the urine. This segment is impermeable to water. The absorption of solutes in the thick ascending limb is more than the absorption of water in the descending thin segment. Therefore, this segment is also known as the medullary diluting segment. Sodium reabsorption in early distal convoluted tubule. Sodium reabsorption in this segment is by sodium chloride cotransporter on the apical membrane. The early DCT is impermeable to water. Absorption of solutes without the absorption of water makes the tubular fluid even more dilute. This segment of the nephron is therefore called cortical diluting segment. Sodium reabsorption in the late DCT and collecting duct. These are hormone-regulated segments. About 3% of the filtered sodium is reabsorbed in these segments. There are two types of cells called principal or P cells and intercalated or I cells. Function of P cells. Reabsorb sodium and water, secrete potassium. Sodium-potassium ATPase on the basolateral membrane actively transports the sodium into the blood. On the luminal side, sodium is reabsorbed via the epithelial sodium channels, ENaCs. Role of aldosterone. Aldosterone acts on both the sodium-potassium pumps on the basolateral membrane, increasing their number. Genomic action of aldosterone and activity. Non-genomic action of aldosterone and on the ENaCs on the luminal membrane, increasing their number. The diuretics acting in these segments are potassium-sparing diuretics. Spironolactone is anti-aldosterone and amiloride acts on ENaCs.